예, 안녕하세요. 수영합니다. 예, 지금부터 에코 글로벌 연차 초청 세미나를 시작하도록 하겠습니다. 오늘 진행되는 세미나는 지속 가능한 환경 기술 개발을 주제로 해외 저명한 연구자들의 강연을 통해서 결과와 신년 기술을 공유하고 에코업 분야에 활동하는 차세대 에코업 인재를 육성하고 참여자 간의 교류를 도모하는 자리입니다. 이렇게 뜻깊은 자리에 여러분을 모시게 돼 영광입니다. 저희가 오늘 총 3개의 강연이 이루어져 있고요. 첫 번째 스피커는 나사 케네디 우주센터의 닥터 아브리 오로크. Am I correct? 예, 오케이. 네, 그다음에 미국 농무성의 김문성 박사님 강연하시고요. 그다음에 휴식을 조금 한 다음에 이제 에이펙 센터의 기후 센터의 이제 전종환 박사님께서 강연을 하실 예정이고요. 그리고 마지막에 단체 촬영을 할 끝까지 좀 가지 마시고 끝까지 사진이 제일 중요하거든요. 증비해서 하도록 하겠습니다. 우선 첫 번째 세미나를 시작하도록 하겠습니다. 저희 미국 나사 케네디 우주센터에서 우주 작물산 책임 연구원이신 아브리 타이틀이 Agriculture Beyond Low Earth Orbit라고 합니다. 주제를 강연해 주셨고요. 우리 박수 크게 한번 부탁드립니다. So Aubrey, your turn.
Hi everyone, I'm Aubrey. I'm here to talk to you about agriculture. That means a bit about the history of crop production. NASA's Artemis back to the moon. Crops will help sustain valuable. Past at NASA's biomass production. Here is arguably that was ground research that led us to growth chamber. Veggie unit, not an advanced. We're also in development hollow growth. That's a vegetable production unit. Plant environment off plant. The future concepts surface system. So a little bit about this history lesson. The biomass production chamber was held at the Space Force side of Kennedy Space Center in what is known as Hangar L. Here they showed the growth of plants vertically, so stacked trays with controlled environment using lighting and hydroponics. They grew wheat and potatoes, and they showed that they could use 20 meters squared of crops, turn over enough carbon dioxide into oxygen to support one crew member. This serves as basis for understanding of how we could grow plants to support crew on the lunar surface. So our technologies that we develop on NASA not only are important for space, but we want them to be translatable back to Earth and research and use on Earth. So NASA initially pioneered the techniques by building the country's first ground facility in a decommissioned hyperbaric chamber from the Mercury space capsule. This innovative approach to farming created the foundation for the industry known as Controlled Environment Agriculture, or CEA. You might know different companies that do this. It'll be in more concentrated industrial areas, in cities, and one such example is Plenty, and we have a picture of one of their vertical farms on the right there. In addition, NASA looked into using different types of lighting that supported crew health so that the lighting wasn't too powerful and bothering the circadian rhythms or the day-night cycle of the astronauts themselves. With this came the advancements of LEDs, and now we know LEDs very much because they're in just about every appliance at home. They're also used for growing plants, and we use that today in our research. When NASA started looking to LED fixtures or solid-state lighting, there wasn't a lot of information and certainly very little standards on their use. So when you develop technology, you may not realize it, but you can become the expert and you can also set the guidelines for their use. So now we use LEDs in plant growth. Some examples of this, we use LEDs to both monitor and manipulate the phenotypes of plants. An example of monitoring is on the left-hand side, where Kennedy Space Center works with the United States Department of Agriculture, Agricultural Research Service, in particular Moon Kim's group, who will be giving a presentation next. And we're using hyperspectral imaging through reflectance and fluorescence to capture the change of the plant after some stresses like underwatering, lower nutrients, or any other type of biotic or abiotic stress they can encounter when they're being grown in space. On the right-hand side, if you look down through the columns, those plants are genetically the same. When they're treated with different types of lightings provided by LEDs, we can change their phenotype. So in broad spectrum white lighting, we might get a plant that's a bit more green with different nutrition properties. In red lighting, they can be a bit more red, and this forces maybe flavonoids or anthocyanins, which are very protective against oxidative stress. And with blue light, we can force that purple color even more, and we can even make the plants smaller in size. So it's very good for manipulating phenotypes when they're all genetically the same. So NASA has objectives. We call this the Moon to Mars. So this is a transition. If we go from the space station to the lunar surface, we hope to learn what we can do on the Martian surface. They highlight objectives such as fundamental plant biology and applied crop production. So fundamental plant biology, understanding how the spaceflight environment affects plants' growth at a fundamental cellular level, but then also applying that to how can we grow crops for human consumption. In this process, we want to define plant species, including methods for their growth, understanding how they can provide sustainable and nutritious food sources for lunar, deep space, and Mars habitation. At a high level, our roadmap looks a bit like this. We conduct studies on the ground to ensure that the plants that we choose are good for spaceflight. They'll go to the International Space Station currently, and that's where we have a lot of our lessons learned, which I'll talk to you about. But in the future, we're looking to systems like Gateway. Gateway is a space station that will revolve around the moon rather than the Earth in cis-lunar orbit. This will transition into lunar surface stays. With the Artemis program, Artemis 3 will be the first attempt after Apollo, since Apollo, to bring people back to the lunar surface, and that'll be the touchdown event to be able to conduct science on the lunar surface after 50 years. We also plan for Martian transit vehicles, so this will be longer stay, but smaller growth areas, a little bit like Gateway, and then larger systems such as Martian surface stay, very much like lunar surface operations. Each of these come with their own challenges because there are different sizes we can grow in and also different environments from each. So why are we growing plants in space? Well, first off, food. But right now we use them as supplemental nutrition because the astronauts have a very balanced packaged diet. So we only really need to introduce new calories through supplemental nutrition with fresh produce. Also psychological well-being. So they've been far from home, far from Earth, among things that are quite silver, maybe a little bit dark in color, and then you end up reintroducing Earth back with some green in the lettuce that they're working with. And so this reintroduces their connection back to Earth. We can also regenerate the atmosphere, turning carbon dioxide, which is quite strong or high on the International Space Station, although it is scrubbed, back into usable oxygen. You can also recycle water. You can send not completely potable water through plants, but instead gray water before processing through plants, and they'll be okay with them when they grow. Okay. So our vision is to ensure food system security on long-duration missions beyond low Earth orbit. So low Earth orbit is currently where we conduct our experiments. That's on the International Space Station. When we talk about beyond low Earth orbit, we're no longer sheltered by the magnetosphere. Magnetosphere protects the Earth from radiation. So as we go further beyond low Earth orbit, we're going to be in a different environment with higher radiation. And when we hit the lunar surface, we'll be with partial gravity versus microgravity. As I stated, our near-term goal is to supplement food on the space station, that packaged diet. Currently, we do not have to reuse the atmosphere or recover water using plants, simply because we use physicochemical systems to do so. Currently, they recycle urine and humidity condensate. The urine, for instance, goes into a reaction with hexavalent chromium. This is reduced to chrome 3. It creates a brine, and the water can be removed, and the other elements stay with the chromium. This purifies about 98% of the water. That goes through a few more filtration steps and then can be drank again as potable water. Long-term goals will be using staple crops to move towards Earth independence. This because we'll get further and further away from Earth, and resupply will become harder and harder. With our research on the International Space Station, we've identified crop production challenges. In low Earth orbit, similar to deep space, where there might be a microgravity environment on that transit vehicle similar to the International Space Station, you have microgravity. The secondary effects of this are the physical effects being lowered fluid movement, so it's harder to water on space station, and also no to low convection. So this means all those little jetties that you have in the air flowing and turning over your oxygen and your CO2 is no longer being used on space station. Instead, we force convection with fans. On the surface, such as the lunar surface, we're going to run into dust from the regolith. So that's very small particles. They can get in crevices and cause problems. And we'll have partial gravity, which actually could aid in watering because it's 1/6 gravity versus microgravity. If we look at the overlap here, we see that we do still have challenges we expect to encounter with watering. We have different devices to provide water. We run into challenges with recycling, so that edible biomass we use, but inedible biomass still exists. Radiation is an outstanding question, which we're starting to address now on space station and will be addressing on the lunar surface. And differences in pressure, because human habitation systems can have as low as 8.2 psi or 56 kPa, and these may still change, but the plants might be more resilient to these pressures, but we ultimately have to use what can be sustained by human presence. At the bottom, if we look at crop and the challenges, we always have the challenge of productivity. We want to optimize biomass. We want to look for stress tolerance so that the plants do indeed grow and provide that nutrition. And then we have environmental optimization, because not only are we off planet in those physical environments, but we can also engineer our physical environment by the habitat we're growing the plants in. Additionally, we have elements of crop scheduling to understand how the plants can help each other and not work against each other when growing. Centrally, we have issues with plant size. We want to optimize the size for the grow space, so this might use engineered smaller crops, dwarf-size crops. We will always encounter high CO2 because this is reliant on scrubbing inside vehicles. As you breathe out, that has to be scrubbed to lower the CO2. Currently on space station, it's about 3,000 parts per million. In this room is about 500, so you can see the difference there. Plants ultimately like about 1,500 parts per million, but when they start to creep up to 3,000, it becomes a little bit more difficult for them to grow optimally. We also consider food safety and microbiome. In the closed environment agriculture sector, what they do is they take a portion of the crops and they'll culture the crops to make sure there's no food pathogens before consumption. Similarly, on space station, we'll grow plants. Some will come back to Earth, some will stay for consumption, but we have to check that equally they are healthy to eat. We're concerned about nutrient output, that indeed we are supplementing the astronaut diet, and this will lend way to actually being the staple for the diet. Sustainability, as I've mentioned, using resources effectively, avoiding abiotic stresses so that we don't stress out the plants as we're growing them, using vehicle resources or the platform we're growing them on sustainably, and then also minimizing crew time, because those astronauts, there's not many of them, but they have a very full schedule and they can't be gardening the whole time. And also being very conservative with our wastes. So plant growth, as you know, has 5 cardinal factors for growth. First off, we want to maintain a proper temperature. We need to provide good lighting, which we do by LEDs. We have to provide water, which is up mass, as we send it to station or recover the water from systems already there. We need to provide fertilizer and proper gas exchange with proper airflow. I've discussed how there's no natural convection in microgravity environment, so that's in low Earth orbit environment. This affects temperature gradients as well as gas exchange, so we force this with fans. When those fans shut off, we can have high humidity and we can have the growth of opportunistic pathogens such as fungus, which we've seen before. We can modify plant characteristics through lighting. Using LEDs, we've downed the requirements for power and we're producing less heat. Fertilizer is heavy, so we know that we need to have something that's a bit more sustainable. And water is difficult because it's even stickier than it is on Earth, and sometimes it can cause hypoxia or the lack of oxygen in the root zone. Okay. So what we look to do in our group, because we do both applied plant science and basic research on plants, is we want to translate research on model plants and crops in low Earth orbit to understand sustainable food production on the Moon and Mars. So I put for scale here, on the space station, we're about 400 kilometers away. And the Moon is about 400,000 kilometers away, and 3 orders of magnitude more just about is Mars. So you can see that this is an issue of scalability. As we get further and further away, we need to be more and more sustainable and better at these practices. So we have hardware on the space station that we've grown various crops in, both veggie and the advanced plant habitat. And we're looking at these plants' adaptation to space. So we know plants began to colonize land about 450 million years ago, and several structures and physiological innovations enabled them to thrive on land. This includes efficient photosynthesis, lignified fortified cell walls, and we see a change in the structure of the cell wall in the microgravity environment, and an interaction with microorganisms, which we're starting to better understand. So these plants have evolved in the presence of gravity. It's very interesting what happens when we remove gravity. In the past 10 years, we couldn't have known it, but sequencing sort of took over. We looked at the genomics or omics generation or era. We've been able to look at genomes, transcriptomes, proteomes, epigenomes, and even microbiomes. And we've characterized what plants are doing in the spaceflight environment. There's so much data, in fact, that we collect it in a repository called GeneLab. GeneLab's a place where you can go online and find all the data from different spaceflight experiments, so you don't have to be a funded NASA researcher to be a NASA researcher. You can go online and look at these data sets and mine them yourself. Below that, we have also the Life Sciences Data Archive. This is where we have metadata and more imaging data that you can take a look at as well, so that might be of interest to students. Overall, we've seen that plants can tolerate the low Earth orbit spaceflight environment. Photosynthesis is not severely affected. They can complete an entire life cycle from seed to seed. We see that phototropism takes over instead of gravitropism. Gravitropism is when a plant orients to gravity. When gravity is removed, a plant can orient to light instead. Microgravity can affect the starch metabolism of a plant, and plants will grow and respond to low fractional gravity. So we expect they'll have a different phenotype on the Moon with 1/6 G and on Mars with 3/8 gravity. So what we want to do is look at spacecraft production through the lens of applied plant science. We understand what the plants are doing through sensors, looking at biological phenomena in space. We understand that cell wall and metabolism are adjusted in the spaceflight environment, and we can engineer accordingly. And we've looked at horticultural approaches, such as whether we should use a substrate or hydroponic without a substrate. And this applies into spacecraft production because we know we need crop health monitoring to lessen crew time. Spacecraft traits can be optimized, such as smaller size, increased nutrition. And then also refining our techniques for growing plants in a small space, such as using vertical farming, or maybe even in the future using in situ resource utilization or the use of substrate from the moon, such as regolith. So the crop candidates that we have so far, we've done a lot of leafy greens for growth periods of 28 days. This is because they're a shorter growth period. You can also pick and eat the plants, meaning you grow your lettuce, your pak choi, your mustard green, you pull off a leaf and another one will grow, and so you can continuously pick from that plant. We've grown tomatoes for 100 days. We've grown chili peppers. Astronauts really like food with a punch because they get a fluid shift, so that affects their taste buds a bit, so they like strong flavors. We've grown radish. We've looked into strawberry, carrot, green onion, and peas. But for long term, we'd look at those staple crops that you might know from the movie The Martian, such as potato, also looking into rice or wheat. That takes a bit more processing and consideration for how we would grow those. So how do we determine what plants go to space? Well, NASA is an engineering-based government entity, so they have what's known as the technology readiness levels, or the TRL. We have made the crop readiness levels, or the CRL. So this goes from 1 to 9. At first, you identify your crop, then you screen for the cultivars that are best suited. You test in relevant settings, which we have at Kennedy Space Center. We have controlled environment chambers. We set those chambers at about 3,000 parts per million, and we grow the plants. We conduct chemistry tests and organoleptics. How does it taste? What makes it taste that way? We do baseline microbiology to make sure that that plant can be sanitized properly and will not be a hazard to crew or systems. We want to make sure that the seed or the propagule for that plant can be sanitized so we're not taking any pathogens up with the plant. We go into flight-like testing, and at a TRL 8, we're at growing in space, and at TRL 9, that plant has been consumed in space. And we have quite a few that have. So what exactly have we done on the International Space Station to date? Well, let me show you some examples. First off, we can't have a meal without setting the table, so we grew zinnia, this flower here. It did quite well. However, the fans did turn off at one point. The plants started to get warmer and warmer, and they started to get a little bit fuzzy. And that was the presence of Fusarium oxysporum growing. So we know that when we have lowered convection, we need to keep that convection going so that we don't run into these scenarios. We have some action shots. So in the lower right-hand corner, they are gracing the table of a meal with some condiments dancing in the background in microgravity. There's one floating in the window looking back at Earth, and another being presented to another astronaut by Scott Kelly for their 300 days in space. We have 2 veggie units, so this is one of the hardwares that you can grow plants on the space station. It's the more simple of the 2 hardwares. You can see that this is a bellows system, so it's a bit rocked forward, but it looks like an accordion, the plastic sides, and then they will go and attach to the top. The reason why they're pink here is because there's an LED platform up top that's shining to grow those plants. The types of plants that we've tested, we call these veg tech demos. So this is Veg 03, red Russian kale, dragoon lettuce, wasabi mustard, extra dwarf pak choi, amara mustard, all looking very nice and happy growing there in microgravity. We have crew engagement. So the crew really like interacting with the chambers. They're getting that connection back to Earth and being stewards for plants and the use of plants effectively on Earth to feed people. They enjoy the process of eating the meal together. And the way that they are sanitizing the crop here is using something called Prosan. They put it in a little baggie and wash them. It's citric acid based. You can also smell these aromas in that cabin. Space station can be a confined environment, and it's probably very welcome to have something like a fresh plant growing. We've shown that you can do transplanting of seedlings. So logistically on the ground, this might be quite simple, but when it comes to logistics of spaceflight, it becomes quite difficult. So here you can see the plant pillow in the background. There's a wick, and this is how we water. The seeds are placed in that wick. It's wetted once. It never wets again, but then the water comes in through the bottom of the plant pillow. You'll have a seedling growing out of the wick. When there's 2 seedlings, you can take one, put it into another pillow, and then you get 2 proper happy adults. Additionally, we've shown the use of seed film. So this is a pullulin, generally regarded as safe polymer. It is originally from a fungus, but you might know it for breath strips. So Listerine, when you freshen your breath with that little piece of paper that dissolves, that's pullulin. And we've put seeds in there. We dissolve it with water, and this helps to contain the plants as the seeds so they don't go whirling around in that microgravity environment. So these technologies are quite a novel, and the ingenuity then transfers into a technology that can be used back on Earth as well. We have radishes growing here, so you can see the little radish bulbs starting to form in the plant pillow. We had space chilies. So this was an outreach project done with a number of classrooms to decide what was the best space chili. And in fact, it was decided to be the Espanola Improved Hatch Chili because that did the best under 3,000 parts per million, which is part of our CRL testing to know that we should send it to space. The crew harvested the chilies. You see the chilies dancing here. And they're always wondering whether your favorite is red or green. Okay, we've grown tomatoes as well, so small variety dwarf tomatoes. This is working towards growing plants that are small for those chambers that we grow smaller chambers on space station. And we can grow them under different lighting, provide LEDs to get different phenotypes from the plants. Okay, so we have many lessons learned from growing plants on the International Space Station. We have ground research to support this work. And now we're looking to beyond low Earth orbit. So up and coming, we want to prepare for larger surface systems. So NASA, in collaboration with the German Space Agency, as well as the Canadian Space Agency and the Italian Space Agency, has taken to growing plants in the Antarctic at Neuermeyer Arctic Station. This is known as Eden ISS, and we have one of our scientists on the right-hand side tending to the crops. So this is a converted cargo container, and it's quite a ways away from the actual habitat that they're working in. So she would have to go out in all sorts of weather to get to this plant chamber, and she was able to understand what it takes to grow these plants. So it is an analog environment for an extreme environment, and we understand there's a lot of maintenance, and we have to schedule for that kind of maintenance when we make plans for things like the Lunar Agriculture Model Ground Test Demonstrator. So this is the round 2 of Eden ISS, and it will be more for simulation of operations on the lunar surface, but on the ground. Inside this chamber, we have planned through workshops to have a vertical farm, a central aisleway. This will have a robotic arm provided by the Canadian Space Agency through Canadensis. That will tend to the crops in the vertical stacks. There will be something like hydroponics. We're trying to understand how frequently we have to reuse those nutrients and resupply those nutrients. The crew actually really like harvesting. So they had hoped that the robotic arm would be more for harvesting, but to the dismay of the student in charge of the robotic arm, he realized he would be doing more maintenance and cleaning. So we tried to improve the things the crew likes to do and take off the other chores that they do not like to do as much. Once the plants are harvested, then they go into the HAB sim. So the habitat simulation is where the plants would be stored and processed for eating later. We also expect that there will be airlocks between the two, and this would be overall a pressure chamber to 8.2 psi. So we used to just have graphics that talked about these things, but right now is the time where we're actually seeing them come to fruition. So it's a very exciting time. We had graphics looking at how could we have a plant production greenhouse on Mars, for instance. This is a depiction of our senior scientist that they made, and he's on Mars growing his tomatoes and tending to his plants. And this may one day be a reality as we test these technologies on the lunar surface. Most recently, our group found out something very exciting, and this is a big step in that direction. Coming soon, we have plants on the moon. The project that we're supporting is called LEAF, or Lunar Effects on Agricultural Flora. at Kennedy Space Center. We'll be doing a lot of the ops and integrating the payload. I'll be in charge of the microbial sequencing and understanding how those microbes change after plants are grown on the lunar surface. So the plan is to prepare this payload, relatively small in size, as solar panels that are driving the power. We grow the plants inside for 1 lunar day or 14 Earth days. The plants will grow until we lose power, but on the 3rd day of growth, a portion of the plants will come back with crew. So the crew will be setting down this payload on the lunar surface. They'll be injecting water and allowing some space crops to grow so we can show how these plants respond to that lunar environment. And again, this is not just one entity's effort. It is an international effort, even working with engineers and biologists from the United States as well as Australia. So it's an exciting time. We're starting to see everything come together that we've learned in low Earth orbit to start doing space crop production and agriculture beyond low Earth orbit. And with that, thank you very much for listening to my presentation, and I'll take any questions you might have.
Okay, so if you have any question, you can use either Korean and English as well. I might translate it, your Korean to English. So, any questions or?
제 생각에는 저는 보건정책학과고 이름 말해야 될까요? 네. 너무 재밌게 들었고 이공학 분야인데도 상당히 흥미가 있다 생각이 되는데요. 이 연구 내용을 들어보니까 이제 다음 단계가 아무래도 작물 좀 영양가나 이런 부분에 있어서 이게 미세 채소라고 들었거든요. 그 채소를 좀 더 맞나? 잘 모르겠지만 채소의 이제 영양분을 좀 고려해야 될 때가 아닌가 해서.
Okay, 거기까지만. Okay, she actually curious about it look very great, but she wondering about the nutrition of the crop. So is that really same with the nutrition inside in Earth as well? So
Yeah.
she wonder, yeah.
They do nutrient analysis when the plants come back. Oftentimes this is elemental or looking for vitamins. Elemental analysis is equal to plants grown optimally in controlled environment agriculture. Also for food safety, they are just as clean, if not cleaner, as produce grown and bought in the market.
예, 이해하셨나요 혹시? 비슷하다고 하시네요. 예. 네.
영양분을 조금 더 이제 신경을 써서 작물을 재배할 때 그런 부분에 이제 앞으로 연구가 이루어지지 않을까라고 이해를
비슷했대요. 예, 그 하는 거랑 얼스한 거랑 뭐 바이라민이랑 뭐 다른 것들이 비슷하게 나왔다고 하십니다. 예.
네, 너무 재밌게 들었습니다.
Yeah,
감사합니다.
she was so impressed your presentation. 또 있으신가요?
Well, I have to say that this is an immense group effort, and I'm representing many people who have done this work. Yeah, so it's a huge undertaking to do plant science in space.
So my question is about the growing of the plants. I couldn't tell exactly from your picture, but are they grown in soil, even in the other environments?
That's a good question. So they're grown in what's known as arsolite. It's a clay material. It's often used for baseball fields. So it is something like substrate based, but because the particles are quite large, it's sort of in between with hydroponics as well, because we're feeding water into that system. In the future, what we're looking towards is something more like hydroponics or aeroponics. And we think that'll be more effective in 1/6G on the lunar surface. There are systems being tested in microgravity, one such as called X-Roots, and they're introducing a small amount of water to the roots and seeing how well they do. But again, microgravity is hard because water really doesn't flow as well and become very sticky and can cause root zone hypoxia.
So to add to that question, sorry. So if you guys are not using soil or plan not to use soil in the future, how are you going to reuse the other biomass waste to get your to use those resources back? Because from what I know, which is very little, you usually use that back in your soil, right?
That's true. So you could make soil through composting. Perhaps you could use that to fortify regolith to become a soil, because right now regolith is not a soil. It does not have microbes. You can use microbes to leach regolith and get fertilizer out of that. But another option for inedible biomass is the in situ resource utilization group at Kennedy not only looks at regolith, but also for using waste to produce gas. So trash to gas method. So you could get some form of fuel.
Perfect. Thank you so much.
자, 하나만 더 있으시면 one more question. 없어요? 예. 그러면 박수 한번 쳐주시죠. Here, Aubrey. Okay, so we're gonna move on to next speaker. 두 번째로는 이제 USDA 우리나라 농무성, 미국 농무성의 환경 미생물 및 식품안전연구소 연구 책임자이시고요. 이 김문성 박사님께서 Automated Multimodal Sensing Technology for Agri-Food Safety and Quality Application의 주제로 강연을 준비해 주셨습니다. 박수 한 번 더 부탁드립니다.
Good afternoon. I can speak Korean very well, but I'll talk in English, if you will. First of all, I would like to thank Professor Cho for providing this opportunity to share our research application in agri-food safety and quality assessment, and also Korea University EcoOp directors and all the involved personnel for making this presentation possible. So I am from east coast of the United States, near Washington, D.C. So we are like few miles from the downtown D.C. area. And we are one of the many agricultural research sites. Our center is one of the largest in the nation, in fact. Before I begin my presentation, a little bit about me. So I see a lot of young people, undergraduate students. How many are undergraduate students here? So many? Just few? So I began my career as an undergrad intern for NASA Goddard Space Flight Center. So I got involved in engineering project as a young person, a young student. And then a few years later, I got involved in remote sensing of studying forest ecosystem dynamics, meaning effect of acid rain deposition. So in the east coast of the United States, there was a lot of factories emitting deleterious gas from smokestacks and whatnot, and it affected the growth of the woody plants in the eastern and Canadian regions. So NASA was using remote sensing tool to assess the effect of the acid rain deposition. So I was fortunate to be involved in integrating sensors on a helicopter platform, as well as support the U2-based hyperspectral imaging missions. And then, finishing my graduate studies, I joined the USDA current laboratory in late 90s, and since then have been working on sensing and instrumentation technology development. So then the laboratory became Environmental Microbial and Food Safety Lab, and about 10 years I've served as the leader of that laboratory, and some of you have been to my laboratory, worked as a visiting scientist as well as visiting interns. So happy to see all of you doing very well in Korea. So as I mentioned, I'm part of the National Food Safety Program. So there are 45 research projects. This is congressionally mandated. That means Congress approved, and hey, you guys at USDA ARS, you conduct this research. There are 45 food safety projects, and the research are conducted all over the country, scattered all over. But by far, Belcherville, my laboratory unit, has the biggest program. So we are more than 10% of the research. So I'm not going to talk about all of these, but some of these research first four topics might be relevant to some of your majors. But today I'm going to talk about sensing and instrumentation. So now you heard Dr. O'Rourke's presentation about spacecraft production, but now we're going to come down to Earth and we're going to talk about food safety and quality of earthbound agricultural products. So food safety, there is no magic silver bullet. So it's collectively, we spend about 10% of ARS budget to conduct food safety in my unit. But little by little, it's a risk reduction effort. There is no one method that gets rid of food safety concerns. So little by little, doing research in multidisciplinary, we are trying to make the risk less for food consumption. So that's the main topic. Now, I'm going to talk about non-destructive sensing technologies today. So let's look at the fruit product, right? Many fruits, grain, meat, milk products. So in a way, you are utilizing it on a daily basis, non-destructive, your vision, right? So your eyes see invisible here. And what do? For instance, you look at fruits. You visually look at it, oh, it looks really well ripe, or oh, it looks like it's got no defects, or it's got some contaminants and whatnot. So in a way, we are using sensing technologies to mimic that so that we can do the bulk processing of food product. And spectral wavelength or electromagnetic spectrum we utilize is from visible and beyond where your eyes can see. So visible to near infrared or shortwave IR. So that's the portion of the spectrum we utilize to assess food safety or quality, in a nutshell. And you hear hyperspectral a lot nowadays. So simply put, you are very familiar with color imaging here. Everybody has color imaging technologies. Your cell phone has color imaging, right? Color imaging is made up of red, green, and blue, 3 filters. So in a way, it's analogous to TV technologies that, before your generation, your parents' generation probably had a black and white TV. Then early 80s, in Korea especially, had a color TV. Now, early 2000, high definition, high resolution TVs. In a way, sensing development is similar to television development. So hyperspectral became available. Actually, it was pioneered by NASA group out in California. So it was flown on U-2 airplane that I showed you in early 80s. So it goes back many years. So NASA develops a lot of cutting edge technologies. But this became readily available, so early 2000, I began to utilize this, and when I joined the ARS, I put together, as an engineering group, laboratory systems to assess or detect contaminants, as well as defects on fruits and vegetables. And as an engineering group, we have developed many laboratory-based systems to assess various food product safety and quality, as well as develop many platforms to utilize it in online or in industry processing setup. So we've developed online automated poultry inspection system and developed a method to look at the entire fruit surfaces using imaging. So if you're trying to assess the safety of any fruit, you just cannot look at one side. You got to look at entire surfaces, right? So you might have some contaminant on apple at the bottom or inside of the stem or calyx regions. So you have to look at the entire surfaces, and we developed the technologies. So past 20-some years, we've developed a number of imaging platforms. More recently, what we do is what's called multimodal, meaning I'm not going to talk about fluorescence, reflectance, or Raman technologies, but we gained know-how. So instead of individual lab-based system, we put together multiple sensing into a smaller packages. And that's what we've been doing past few years. And also, Dr. O'Rourke talked about space ag, right? So NASA has already invested a lot of resources and funding to grow plants in space. However, what's lacking is, a good question was raised earlier, that how do you know it's nutritious, or how do we know that it's growing optimally? So there is some sort of sensing needed. So about 5 years ago, Dr. O'Rourke's group and our group began to collaborate and trying to utilize hyperspectral imaging technologies to assess plant health, as well as assess the safety of the crop production in space. So it's very early stage, and a lot of teamwork, a lot of manpower is involved. I mean, it's not just two persons doing it. Dr. O'Rourke's group have over 10 people, and my group has over 10 people involved in this, including student interns. So I like to emphasize teamwork is very important, and you cannot do it by yourself. So I'm not big on words, so I'm not going to read this, but now what's the recent trend? So as I mentioned, we've developed many imaging or sensing platforms, but it's not really easy for untrained people to use. So we're trying to make it automated, meaning you don't have to have a specific training to run your laboratory equipment. So, so any common people can hopefully use it. So trying to make it easy. So that's the really challenging part, right? For instance, you download an app, and if you have to read a manual to run an app on your smartphone, you're probably going to delete it, right? You wanted to download it and I can figure that out. So that's kind of the inspiration, and we are trying to make these sensing systems as easy as possible for end users. So today, this is engineering project's objective for next 5 years, but we're going to talk about UAV drone technologies. So some of you may have flown drones, but for research purposes, we are not just using a single color imaging camera. It's got more than single camera, so I'll share that with you. And two other things: handheld for contamination and sanitation inspection, and for SpaceEgg hyperspectral imaging platform. So I'll talk about those three. I may be going a little too fast, so I'll slow down a bit. So once again, this is pictorial depiction of what I'm going to talk about is autonomous UAV system. Hopefully, when we are done with this project, it's still ongoing project. So we are still developing it, but I'd like to share with you what we've been doing, what are the challenges and whatnot. So a system that can autonomously go and get the information you need. And second is handheld devices for contamination and sanitation inspection. So this is a commercial prototype we developed. And lastly, the collaboration effort with NASA Kennedy Space Center, the SpaceEgg crop monitoring system for plant health, as well as any safety concerns involved in growing crops. So before I begin, why we are using, and Dr. Jakub Peczewski is in the audience there, and he has been using drone technologies to assess water quality. So in the US, we use irrigation ponds, something like this, to use as a irrigation source for huge crop fields. So his research is to assess the pathogenic bacterial survival and population in irrigation pond. And this one has about a 1-minute video, so I'll share that with you. The volume is really low.
On a pond near Chesapeake Bay in Maryland, USDA scientists take samples to test for E. coli, which indicates the potential presence of harmful bacteria. Overhead, they use drones to capture images from the same spots. Later in the lab, they'll analyze the sample data and images to identify which parts of the pond to evaluate for potential crop contamination from irrigation water. Jakub Peczewski leads the team from the Agricultural Research Service.
What we are doing is helping to figure out where, when, and how to take samples. And eventually we are pretty sure that farmers will use this technology.
Farmers who need a simple, affordable way to test irrigation water quality could benefit. This experimental drone captures multiple images.
Our drone is equipped with 5 different cameras. One of them is a multispectral camera, which does 5 different wavelengths per shot. And what we can pick up on the photographs is something we just can't see with our own eyes.
From the boat and from the shore, the team samples in multiple areas and depths.
We will typically pick anywhere between 3 to 5 points, and at each of those transects we will do 4 to 5 different depths.
In the future, drones may also collect the water samples as well as images. The ARS researchers hope this technology will help farmers save time and money while promoting food safety from farm to fork.
We want to make sure that whatever the farmers are required to do, that it's not going to put a strain on them. So this may be an easier way of them doing that by just flying a drone over and seeing what areas should be sampled.
In Queenstown, Maryland, for the U.S. Department of Agriculture, I'm Pat O'Leary.
So this was done about 5-6 years ago, led by Dr. Jakub Piechepski, who is sitting over there. So if you have a specific questions, please go after him. Anyhow, so it's a collaboration with his group and my engineering group. We wanted to leverage hyperspectral, thermal, 3D scan, as well as RGB. So we wanted to put more sensors on a UAV. And also, bottom line is you hear a lot about AI and machine learning. So we wanted to have edge computing, meaning instead of downloading the data and post-processing it, we wanted to embed the model and also small board computer to process it in near real time as it's collecting imagery. So it is really challenging project to accomplish this within 5 years. At the time my team was proposing this, we didn't realize how challenging this is. The reason being is, this is typically how many people you need to, fly just a hyperspectral based, drone. Doesn't even have all the other sensors I just mentioned. Just simply 7-8 people spend about a whole day, and you barely capture one scene images like this. So this was done with Jakub Piechepski's group, and my group went out to the field, about 7-8 people like this, and collected this imagery. And small footprint here is about 20 meters by 100 meters. It's already 10 gigabytes. So you start, collecting large areas, you're going to deal with hundreds of megabytes of data, and you got to process it. It's really challenging. So it's best that we collect the data and trying to process it in real time on board and ditch the data we are not using. So that's where we are going. And I'm sure that some of you have flown a drone, small miniature drone or whatnot. So for color imaging, it's straightforward. You fly it, and image processing is your vision, right? You can do pretty good job of differentiating different features of a landmark, but trying to model based on imagery is more challenging than that. So a lot is involved, and so it's a work in progress. So why we are using the drone for? One is for, as you saw in the video clip, trying to model water quality of irrigation pond. Second is we wanted to inspect produce field for animal intrusion, because if the produce field is intruded by animal species and they have fecal materials or droppings, then industry doesn't want to harvest that produce field. And currently, as I mentioned at the beginning, you go to grocery store, you visually inspect as a non-destructive. So it's still done by human inspectors visually. So we want to automate it. So that's why we are developing this. And so look at current commercially available hyperspectral images, bulky and big, but we are identifying miniature components and also onboard computer. So this weighs about 50 grams, another RGB camera, 95 grams, and hyperspectral imager is 240 grams, and altogether, total weight is about 385 grams. So the lighter the payload is, longer you can fly the UAVs, and you can cover large field area. So that's another engineering challenges. And once again, multimodal meaning color, hyperspectral thermal, as well as LiDAR 3D scans. So we are combining all this and working toward, this is an end product. So simple graphic interface. You fly out your drone and it'll tell you where the problem area is in case of irrigation water. Embedded model will tell you that this irrigation pond has some microbial and E. coli loading issues. So that's the end result. And the key here is using NDVI and a small board computer on board. All current technologies, they just capture data and you download it. But we want to process it on in real time. So it's going to take some time, but little by little, we're going to test out the first system this summer in collaboration with Dr. Pachevsky's group. So hopefully next time I come present this, we'll have some preliminary results. So I'm going to move on and go to handheld imaging devices. So think about why we are doing same thing for like over 20 years. Just like your iPhone was Samsung Galaxy versions, Galaxy 1, 2, and now it's what, 24, 25? So things get better. So we can't just do a research 1 year and be done with this. So this is a prototype system, and we are still doing this. And what this handheld imaging device is for, it's a contamination and sanitation inspection of food processing facilities or meat inspection and whatnot. It's all based on what's called fluorescence-based imaging system. So once again, it's a non-destructive. And also this is a U.S. auditor auditing food serving facilities, and requirement is you cannot touch any surfaces because of the cross-contamination concern. So this was tested several years ago. This is an earlier version. And so he would go to kitchen area and illuminate it, and it'll tell you if there's some contaminants and shows up as a bright spot. Image on the right, this is inspection of a meat. So slaughterhouse, you slaughter a bovine, and when internal gut is eviscerated, the fecal material splatters onto the meat, and bovine fecal material is sources of E. coli. So using these handheld imaging devices, it'll tell you where the spots that's got fecal smears on piece of meat. So this has been tested in the lab for many applications. But the problem is it doesn't do automated detection for the end user, meaning I give that unit, if you have enough time to inspect it, you can use it. But things move fast in real world. I mean, when they slaughter a meat, they don't wait for you. They're gonna be on they are hung on a shackle, and it moves fast. So you got just few seconds to inspect it. So it's really challenging to use this kind of devices if it doesn't tell you, hey, there are something that's has contaminants on it. So I'm going to go back here. So it's very important to meet up with the commercial partners to be able to develop a commercial version and be able to incorporate the latest electronics as well as LED technologies. So for about 15 years, we developed internally, and then with a joint partnership with the commercial partners, we developed the commercial prototype, 2 versions here. So it's got some improvement on it, but bottom line is, so this is our commercial unit 1 that was developed a couple of years ago, and it's showing what this water faucet looks like, but it highlights where the contaminants are, or some sort of splattered material. So that's what imaging system picks up. And also, a lot of kitchen areas needs to be inspected by inspectors. And once again, you cannot touch any surfaces, because you go into a facility as an inspector and you start touching surfaces, the facility owners are going to claim that you came in with bacteria and cross-contaminated. So any food safety audit you are not allowed to touch any surfaces. So this is what the new instrument sees, but how do you know this is, red or yellow spot is bad? So that's where we need to utilize image processing and machine learning. So this is fecal contamination detection on meat, automatically telling and annotating where the contaminant is. So this is, I believe, beef samples. Inspectors pointing where they can see visually. So current inspection requirement is that only visible fecal material needs to be removed. However, sometimes these machine vision can see better than what human can see. So based on machine learning AI, you can also annotate contaminant spots where your eyes cannot see. So it's got both capabilities, and the commercial unit will be available soon. So this is second generation of the imaging devices we've been developing in collaboration with this commercial partner. And this is the last section, and I think I'm going too fast. I got till 3 o'clock or 2:50. Yeah, okay, sorry, I thought I had plenty of materials, but maybe I'm speaking too fast or not covering everything. But I don't need to explain again the NASA's effort to grow plants in space. I mean, this is a International Space Station system that NASA has flown and grown samples and leafy greens. And they are doing extensive research using their growth chamber. So in collaboration with NASA Kennedy Space Center, we develop hyperspectral imaging devices that is capable of reflectance as well as fluorescence measurements. So it's in our laboratory, putting together the system to be installed at NASA Kennedy Space Center. As I mentioned, this is constantly ongoing, and as soon as we see something better, we are making an improvement. Just like iPhone or Samsung smartphone, every year they have new versions. So just like commercial units, as researchers, we have to stay on top of things, and if we see and we found something better, we have to enhance it or improve it. So in terms of LED-based illumination for imaging, we are constantly looking for better systems. And if this ever makes to space, we have to make it really light and compact. I mean, constantly we are thinking about how can we make it smaller and lighter, but works better. So that's the challenging part for us as an engineering group. And so this is a prototype system. How it works is it's a three-dimensional gantry system. So it moves back and forth and captures reflectance and fluorescence images. So in a lab, we programmed all these linear tracks and sensors to make it work. So that's kind of what engineering group does to develop this kind of system. And this is being tested at Kennedy Space Center here to the right, the initial installation of the system. But so it's a fully automated, collects the data, and Kennedy Space Flight team, led by Dr. O'Rourke here, they collected a lot of data. I mean, trying to prove that sensing works for assessing plant health. You cannot just run one experiment and be satisfied. You gotta collect replicates of data. So that's what they have done. I mean, this is Dragoon plant with a mild water stress, and so far amount of data we've received is 4 terabytes. So it's just 1 year experiment, and think about 4 terabytes. I don't think anybody who's captured the images or pictures even come close to 1 terabyte. Has anybody? Your backup disk, do you have a terabyte? No, that's the amount of data we are collecting to develop a model to make sure that we can utilize this information. So we've just done some preliminary analysis, and water stress is most important factors in space, because you cannot take unlimited amount of water to space. So initially, NASA team has subjected these plants to mild water stress. I mean mild, because not severe enough that it'll kill the plants at the end, but however, the idea is to automatically detect any water stress early in the process, so you can rectify it or correct it if it needed more water or whatnot. So it appears that utilizing this sensing, we can detect it at very early stage of the water stress conditions. So we still have to process a lot more data, but it's, as I mentioned, team effort. NASA team is still collecting additional data, so pretty soon it'll double from 4 terabytes to 8 terabytes. And we are setting up high capacity computers, servers, to analyze this huge data. So that's what we are doing at this stage. So in a nutshell, I share some of the research we are doing as an engineering group. And so you hear something about Space Ag, but at ARS, USDA ARS is a research organization. So USDA has many entities, inspection services, and also rural development, as well as NIFA, grant funding agencies. But ARS is an in-house research organization. So that's, in a way, what we do in terms of food safety and engineering. So, well, here's a preliminary image processing result. So within few days, the Dragoon subjected to mild drought or water stress. You can visually see clear differences between control and whatnot. As I mentioned, this mild water stress is not killing the plants. It'll still grow, but what's optimistic is we can possibly use sensing technologies to be able to tell pre-visual, meaning your eyes cannot tell whether it's got any stress conditions on the plant surfaces, but sensors can tell that these plants are being subjected to water stress, so alarm maybe astronauts, hey, do something about it or pay attention. Because once you lose this crop production, you know, you are in trouble in space, right? You cannot just go to grocery store and buy it. So this is one of the critical research, but I wanted to re-emphasize, like, human nutritious food production and stuff. This is a huge team effort. So what you're seeing is just tip of a needle, you know. So and what's next for us? So we have additional funding, and it's in collaboration with, once again, Kennedy Space Center and North Dakota State. They have a great aviation program, and they have also mock up Luna habitat. It's called, I forgot, inflatable Luna habitat, and they train students to live in there for 2-3 weeks, and you get a little certificate. So, and they have this vertical farm set up. And they asked us to develop a monitoring devices. So this is a conceptual design, and we are going to put together this by end of this summer, and we'll travel to North Dakota and install it here so that student can use it to kind of get a taste of what it feels like to do space farming. So a lot of interest in space farming, and I heard that Korean government is putting together agency or department to do space ag. So timing is right, and now you heard from Dr. O'Rourke and me about space ag and space farming. Maybe it'll interest you or get you excited about this and get involved in this, you know, new venture endeavor for you guys. So, as I mentioned, team members, Dr. Jakub Peczewski leads a project, and these are the scientists and many postdocs and students. And also, it's very important, it's a teamwork. So, we collaborate with a number of entities in the US as well as Korea. And So Korea University, few of them, and Professor Cho, I intend to open an opportunity for intern students to come visit, interact with us, and show our facilities, and hopefully you can think about your future goals and be involved in science, research and development, and engineering project and whatnot, because many opportunities. So with that promise, that's my presentation. Thank you very much. Anybody?
It's a really interesting topic. 영어가 불편해서 갑자기 예, 편한 한국말로 하겠습니다. 그러면 예, 저기 계속 발표 중에 갤럭시 뭐 아이폰이 계속 그 버전업이 되듯이 저 프로토타입 뭐 핸들 뭐 something 예, 그게 계속 그런 식으로 업그레이드하는 식의 리서치가 필요하다. 또
예.
그걸 몇 번 그 반복을 하셨거든요.
예.
그래서 아마 제가 생각하기에 그 혹시 그 연방정부 뭐 세니러를 뭐 컨빈스하기 위해서 예산을 계속 따기 위해서 그런 얘기들을 과거에 했어야만 했던 적이 있는지 궁금합니다. 사실 여기서도 이 가버먼트 쪽에 일을 하다 보면 옛날에 했던 거 같은 거 아니야? 뭐 이런 식의 얘기를 많이 들었거든요.
저도 한국 정서 고등학교 때까지 끝나고 갔었기 때문에 그런 정서를 잘 이해를 하는데요. 미국 연방 정부 연구하는 거는 장점이라고 하면은 그러니까 여기 대학에 계신 분들의 애로 사항은 뭐냐면 이제 정부나 어디 기관에서 펀딩을 받아오면 5년 내에 다 끝내셔야 되는데 그 연방 정부 리서치 할 때는 지속적으로 할 수가 있어요. 그렇다고 해서 똑같은 거를 계속 하는 게 아니고 아까도 말씀드렸지만 5년마다 우리가 향후 뭐 할 건지 과제를 내서 그 블라인드로 피어 리뷰를 받아와서 허가를 받으면은 하는데 그 중에 하나가 뭐냐면 이건 엔지니어링 프로젝트고 기술도 항상 향상되고 개발되기 때문에 우리가 지속적으로 해서 더 나은 그 퍼블릭을 위해서 연구를 해야 된다고 해서 지지를 받을 수가 있어요. 그리고 저희 같은 경우에는 미국에는 라비가 통하거든요. 그래서 우리가 같이 일하는 회사들이 라비를 해줘가지고 증액도 받아요. 그러니까 한국하고는 조금 시스템이 다르죠. 여러분들 들었지만 미국에서는 라비스트가 있고 그게 정식적으로 통한다고 그랬는데 우리 ARS에서 그거 두 개 제일 잘하신 분이 야쿠 박사하고 저였어요. 그래서 지난 4년간 증액을 500만 불 받았어요. 그러면은 이 연구하는데 500만 불이라고 하면은 70억 원씩이잖아요. 그러면 그 돈으로 이런 기구들이나 이런 걸 더 할 수 있고 합법적으로 그렇게 라비 받아서 증액을 받아가지고는 연구를 계속 하고 있습니다. 그리고 포스닥도 늘려가고 있고 또 뭐 그런 식입니다. 좋은 질문 해주셨습니다. 좀 정서가 다른 것 같습니다. 여기 저도 여기 보면은 한국 정부기관이라고 일하지 않습니까? 농진청이나 그 농관원. 근데 거기서도 내년까지 뭐 실용화될 실질물을 내야 된다고 그랬는데 저 같은 경우에는 조금 우리 한국에서 연구를 지원해 주시는 분들이 그 마인드셋을 좀 바꿨으면 좋겠습니다. 그리고 제가 한 10몇 년 전에도 3M 그 Quality Assurance People하고 만났는데 자기네는 PhD 받은 과학자만 500명이래요. 근데 거기서 상용하게 나오는 거 기대하는 치가 회사 측에서요. R&D를 하면서 15년에 하나래요. 그러면 그게 진짜 선진 그 3M 하면 큰 회사 아닙니까? 거기에 마인드셋인데 어떻게 학과나 뭐 이렇게 조그만 연구기관에서 사이언티스트 5명, 10명 해놓고 뭐 실적을 내고 그러니까 그런 정서가 조금 제가 볼 때는 좀 답답하다만 애석하다고 표현을 해야 되나요? 그러니까 그런 거는 조금 임프루먼이 있어야 될 것 같아요. 근데 어차피 항상 그 총괄 책임자분이 바뀌시고 그러면은 당장 뭐 보여줘야 될 게 있고 그러니까 그 정서는 확연히 다른 것 같습니다. 그래서 예, 좋은 질문 해주셨습니다.
예, 어쨌 뭐 아시겠지만 내년도 R&D 예산이 동결됐다는 아주 희망적인 얘기. 아무튼 뭐 상황이 쉽지 않은데 미국은 펀드가 계속 늘어나고 있고 굉장히 부러운 상황인 것 같습니다. 예, 혹시 또 질문이 있으신가요? 혹시 저기 영어 잘하는 학생,
네,
한국말도 잘하죠?
저 영어가 조금 더 편해요.
오 진짜.
English
Sorry,
is fine.
Yeah, I'll ask
okay.
my question in English. So I saw that it was a really interesting presentation, and I'm not really that well versed in this area, but I was just curious, what are some ways that we can use this technology more commercially? Like I did see a bit of like how it was going to be used in NASA and stuff like that, but obviously I know that tech is like you can use it in various ways, and I thought what is your take on what would be most commercially useful for it?
Well, for instance, one example is this handheld imaging devices. So we are research entities, so we cannot commercialize stuff. That's why we are teaming up with a private company like this. And what I can do is make sure that this technology is available for public or food processing or prep facility can get a hold of it. And so constantly we are collaborating to make sure that it can be used in broad applications, right? And this will be available commercially. So already NAQS in Korea, they are interested in utilizing this for their inspection program, But this kind of technologies wouldn't be available to individual persons because unless you have a $15,000-$20,000, right? So, however, we work for the public. So, anything that enhances safety of food safety of for public consumption, we really work with these kind of entities to make it available to enhance food safety of the U.S., not only U.S., international. And one thing nice about working for U.S. government, as Aubrey mentioned, they have so many international collaboration, and so do I. As you can see, I used to have more European collaborators, but now a lot of visiting scientists, postdocs, they all went to these schools. So I have to collaborate. In a way, I have to collaborate with them. And Korea University isn't any exception. So I intend to collaborate. And as a student intern, you come visit us, then I'll make sure that you get to see our laboratory and get you involved in this R&D efforts as well.
Thank you.
예, 시간이 뭐 좀 남아서 혹시라도 또 있으신가요? 질문 혹시 뭐 그래서 제가 마지막으로 질문 하나 드리면은 멀티모달 하시면 드론의 멀티모달이면 어떤 센서들이 이렇게 해가지고 하시는지 좀 궁금했습니다.
멀티모달이라고 했는데요. 지금 그러니까 칼라 영상도 하고 초분광, 그다음에 thermal and 3D scan. So we wanted to combine all these as a one package, because it turned out actually something I didn't mention. It's slightly better, but so this is combining both reflectance and fluorescence and combined, meaning we are using both reflectance and fluorescence. Preliminary indicates that you use multiple sensing system. The results are slightly better. So we still need to test more data, but so in a way, that's why we wanted to utilize multimodal sensing.
그러면 뭐 이렇게 멀티모달이니까 좀 AI나 머신러닝 모델도 같이 커플링돼서 하시는 거예요. 알겠습니다. 예, 혹시 또 질문이 없으시면 김우성 박사님께 박수 한번. 예.
감사합니다.
자, 저희 휴식 시간 할 건데 브레이크 타임 할 건데요. 제가 어나운스먼트 되게 중요한 게 현장 곳곳에 QR 코드가 적혀져 있거든요. 있다는데 저기 뒤에 있나요? 예. 입구 쪽에 QR 코드가 있는데 그걸 하시면 10,000원 상당의 기프트콘을 증정한다고 합니다. 네, 저도 해야 될 것 같긴 한데 어쨌든 so 20 minute break time and we're gonna start the sessions 오케이. 몇 시지? 하면은 2:20. Okay, we're gonna start again the session 2:20. Okay? 2시 20분에 시작 다시 하도록 하겠습니다. 죄송합니다. 3시 20분. 3시 20분에 시작하도록 하겠습니다. 예. 그 발표 그 마지막 세션 끝나고 단체 사진 한번 저 증빙이 있어야 돼가지고 단체 사진 한번 찍을 건데 꼭 가지 마시고 사진 찍고 가셨으면 좋겠습니다. 아시겠죠? 예. 우리 마지막 세션 마지막 순서로 에이펙 기후센터의 전종환 박사님께서 발표를 해주실 텐데요. 그 Territorial Ecosystem Carbon Dynamics and Global Climate Corporations, a Case Study with APEC Climate Center라고 하는데 저희 그 기후 변화와 관련해서 국내에서 최고 대가이신 우리 전종환 박사님 모시게 돼서 너무 영광으로 생각하고 있습니다. 예, 우리 전종환 박사님 박수로 한번 맞아주셨으면 좋겠습니다. 예.
Thank you, Professor Joe. Yeah, I'm not that big guy as well, actually. So, rather than that, I'm a humble scholar. So call me a humble scholar. Yeah. And yeah, hi everyone. First of all, thank you very much for joining me today. I'm Jong, working at APEC Climate Center. It's a short, is APCC. I'm very pleased to present terrestrial ecosystem carbon dynamics and global climate cooperation. Our focus will be on a case study, is a GCF project. Okay, this is the already discussed in this presentation. Any of you who doesn't know very much the APCC, I will briefly introduce the APCC, and we'll discuss carbon cycle in terrestrial ecosystem, and we'll give some examples or some application of land information system, which NASA play a leading role to develop. And finally, we'll introduce some GCF project, the targeting the Vanuatu, one island nation. Okay, APEC Climate Center, APCC. These color-shaded countries are member of APEC. It's located around the Pacific Ocean. And you probably know the USA, Canada, Mexico, Peru, Chile, Russia, China, Korea, and Japan, and Australia, New Zealand. Or the one Pacific Island country is the Papua New Guinea here. Okay. In 2005, there was a summit meeting in Korea, and the 21 summit unanimously agreed to establish a climate center in Korea. So what we do is basically we are doing some scientific research on climate variability and change throughout, especially for the APEC region. So the basic role of the APCC is we are collecting global climate forecast from the 15 institutes, including NASA, one of those, and BOM, the Australia Meteorological Office, and even we have some couple of some European institutes too. They are not APEC member economy though. And so the basic role is we issue monthly rolling 6-month MME climate forecast. Basically, the MME forecast is higher performance than any other single model. This is some scientific proof. And for the terrestrial ecosystem carbon dynamics, so yeah, the left-hand side, this is the Earth system. So we can see land, ocean, some atmosphere. There are very complicated and sophisticated interactions between these three components. So in the model consists of these 4 components too. So this is one coupled model. It's called GLOW C6. The UK Met developed this system. The UM for atmosphere, JUICE for land, and NEMO for ocean, CIS for sea ice. And these 4 component models were coupled with this OSC's a coupler. Okay. because this is really big model, and we have to handle lots of data set because we handle whole globe. So that's why we needed a supercomputer system. So currently we use this system. It's called Naru supercomputer system. It has more than 600,000 cores, and the theoretical speed is 51 petaflops. Honestly, last night I struggled with some code error on supercomputer, so I slept very late. That's why I'm really tired today. But I will do my best. Yeah. And, this, even though we have really, really complicated and sophisticated interactions between the land and atmosphere, but simply speaking, we could see only these three: energy, water, and carbon. But today, as we mentioned before, we focus on carbon. Something complicated here, but let's say more simply speaking, it's just three components: photosynthesis, lithophore, and soil respiration. Okay, so if we just look at land and atmosphere, okay, we have three systems basically. The first one is, let's say if photosynthesis and respiration are equal, there is no change in carbon dioxide concentration in atmosphere. But the higher photosynthesis, maybe the concentration will decrease. And then the higher respiration, this system, the carbon dioxide concentration in the atmosphere will increase, right? This is a very simple math. So keep in mind that this is what we say. So we, generally speaking, we just said, okay, land or terrestrial ecosystem act as carbon sink because the photosynthesis is much higher than respiration. But what if the photosynthesis is less than respiration? Then even the ecosystem will act as carbon source. So recent research shows something similar things. Okay, the land may act carbon source because if you look at this slope, this is the ratio heterostrophic respiration to the total soil respiration. You don't have to worry about too much. Let's just look at this result. Yeah, it's very simple. And yeah, the graph shows the slope increase, the significant increase. It says from 54 to 0.63. It's suggesting that soil microbes become more active due to climate change. It may imply that the role of the land surface as a carbon sink, that's what we think so far, possibly weaken. So this process may very kind of some dark, the future things, man. So we have to stop this kind of the climate change. Otherwise, we will meet the climate cliff. We cannot back at the point. We cannot back to the current system. That's what we call the climate cliff. This is how work the why we happen these things. Basically, the higher carbon dioxide concentration will heat the atmosphere. The higher temperature of atmosphere will heat the soil system. Then the warmer system will give you the microbes more comfortable. That's why the microbes are active more actively. That's why we will give more carbon dioxide concentration to the atmosphere. And it makes worse and worse. So we have to stop this kind of cycle. Okay, this is how we measure the photosynthesis. I was surprised when I worked the USDA, the park area. The human beings can measure photosynthesis. Before I joined the park area, I used to work only the engineering research area to collect water samples and to analyze the samples, those kind of things. So I never thought about the people, human beings can measure this photosynthesis. But anyway, we can measure that. This is the facility located in Bassville, Maryland. We can very accurately control the concentration of CO2 or temperature and humidity inside this chamber. So let's say we raise the carbon dioxide CO2 concentration doubled, then this current ambient CO2 concentration, so that we can see in the future our crops will grow properly or not, something. Okay, we can do research those kind of using this facility. And this is the portable system, but honestly, this is not portable. This is really heavy, so it's really hard to can handle these things. But anyway, it's called the portable system for photosynthesis. These tubes are where we measure the carbon dioxide efflux from soil surface. Okay, this is a simple one. It's called the standard chamber method. There is something like a cap. Inside the cap, there is a CO2 sensors. So just insert the already installed this ring, and then as time goes, probably the CO2 concentration will increase inside the chamber. The slope will represent the CO2 efflux. That's how we measure the CO2 efflux from the surface. And this is more directly measure CO2 concentration on soil. Okay, we install these Vaisala CO2 sensors, the certain depth, different depths, and so easily we can calculate some gradient of carbon dioxide concentration. Last column will show the modeling part. So we cannot measure the whole globe, this carbon or some CO2 concentration or effluxes, because that's why we need this modeling approach. Okay, there are two types of modeling, the carbon pool, and the other one is the process-based model. We will not discuss in detail today. Yeah. This some result. When I was in the Johns Hopkins University, I installed some these kind of sensors, Baltimore suburb, Maryland, and to see some differences in carbon dioxide effluxes from forest and grass. This, let's say the red one represents forest, the blue one grass. So the carbon dioxide effluxes from grass is much higher than the forest. I think the soil temperature will play a role for the region, the difference, I guess. This total measurement is covering the entire year, October 2010 to October 2011, and we can see clearly some seasonality. Growing season is the carbon dioxide much higher than the other season, as we can easily expect. And this is modeling result. The model clearly well estimate the carbon dioxide effluxes from each land cover. And I brought some applications of land information system. It's called just LIS. Land information system. So NASA play a leading role to develop this system. So inside the land information system, there are dozens of the land surface models, which can simulate whole land surface fluxes and states, like soil moisture or some evapotranspiration sort of things. And So currently, we using the cellular images and data simulation, we can update some states, or through the optimization and uncertainty estimation technology, we can calibrate the parameters more accurately, so that we can get more accurate result from this land information system. Anyway, this land information system applications are lots of the areas, the water resources, some weather, the agriculture, some extreme weather, and public health. And I heard that even the military sector, they use the land information system. But I don't know what or how they use, but that's what I heard. And this is the some comparative assessment of energy and water budget. We selected 7 major basins over the world. This is some Asian region. This is American. The Mississippi and Amazon and Murray-Darling Basin in Australia. We selected 3 land service models: NOAA, MP, and JULES. Let's skip it in detail. And we collected some cellular-based terrestrial water storage, and some evaporation, also some energy budget, energy components also we collected to estimate or access the model estimations. Okay, some generally speaking, these figures demonstrate some low performance in the water component for the semi-arid regions, while highlighting the low performance in energy component for the tropical rainforest region, specifically the Amazon basin. I think the low performance probably resulted from some low availability. So for the water component, the semi-arid region, in the region there is low availability in water. Similarly, the energy availability is lower in this rainforest region. This figure shows the different types of droughts and how they developed. Okay, one meteorological drought, it starts with a lack of rain to agricultural drought. This happens when the lack of rain causes the low soil moisture, which affects the crops mainly. And 3, hydrological drought. It occurs when the low soil moisture and rainfall reduced the water levels in rivers and lakes, leading to low discharge and groundwater storage. Okay. And 4, socio-economic drought is the final impact, where the human activities and economies are affected due to the these previous droughts. Okay. The main causes are the anomalies in precipitation temperature. Mainly this is impact the human society. And as a hydrologist, I focus on the soil moisture. So we developed the soil moisture index using the land information system applications. And this is the category of the droughts. And this is the result we developed the soil moisture index for the East Asia region. For this bigger region, a large region, we cannot get this field capacity or what are the wilting point, the 2 critical parameters to get this as soil moisture index. So this is a typical way to estimate the field capacity and wilting point. For the field capacity, we assumed as 95% percentile of soil moisture, and the melting point for 5 percentile of soil moisture. Okay, let's move on the some application of the GCF project. This is more on the adaptation project rather than mitigation. So it's not directly dealing with the carbon. The English title is Climate Information Services for Resilient Development Planning in Vanuatu. But the Venkira is in Bislama. It's Vanuatu official language. Is more popular for the project name. And Vanuatu is the island nation located in the South Pacific Ocean, east of Australia, and northeast of New Caledonia. The country consists of approximately 83 islands, and its population is about 300,000 people. You may think about some Iksan city in South Korea. And this country is divided into 6 provinces for administrative governance. Torber, here Samma, Prema, Malampa, Shepa, Tafea. In the Shepa there is a capital called Port Vila, and this island is called Tana. If you visit Tana, you can see the active volcano, very in the crater. So I visit last month, I guess, yeah. Really, so you can hear the volcano sound, even you can see some flame if you visit the nighttime. Yeah, it's really exciting, yeah. Experience though, yeah. Anyway, the project enhanced the ability of decision makers, communities, and individuals to effectively plan for and respond to the impacts of climate variability and change. It focused on 5 key sectors: agriculture, water, fisheries, tourism, and infrastructure. By strengthening planning and response capabilities, we aim to address both long-term and short-term climate challenges through advanced climate services. Okay. So the GCF funded about 18 billion US dollar. Actually, the APCC's activities was over last year, but the project is ongoing to install a single radar system in the Vanuatu. And our delivery partner is the CSIRO and BOM. They are BOM is the governmental, the Institute of Australia. CSIRO probably some research institute in Australia. And VNGD is the Vanuatu's government department. Yeah. SPRAVE is the accredited entity of the kind of regional institute. Okay, yeah. This slide introduced OSCAR, the short for tailored system of climate services for agriculture. We developed this web-based system. This system basically is a comprehensive solution aimed at enhancing agricultural productivity through advanced climate services. And we can get some weather information and farming advisory based on crop modeling, and some early warning information as well. And finally, digital soil map ever in the Vanuatu. Okay. Before the OSCAR system, there is only paper version of a soil map in Vanuatu. We digitized the soil map and provided the kind of digitized everything. Okay, we believe this system leverages modern technologies to provide some enhancing the decision making, and yeah, through the tailored climate services. There are 2 types of OSCAR system. Left one is web version, right one is the mobile version. So you may think, okay, some developing country, the smartphone, maybe you may, anyway, even the developing country everybody has their own smartphone. And that's why we developed this mobile version. Okay, the web version. If you are a normal user, then you don't have to log in to use this system. But for the government employee or some experts, they need to log on to use some more sophisticated functionality. So for the mobile version, the left one is the normal version with very fancy graphical information. But think about the developing country, the internet connectivity is very low and very, very poor. That's why we developed this text version only, so that we can get much faster data transferring. Okay. Of course, even the picture shows only English, but this system is serviced in Bislama too. Some deep learning technique was applied to get this offline digital map. So we developed the 6 provinces for this OSCA system. Basically, we use the Sentinel to true color images. And enhanced deep super resolution was applied to downscale 4 times from 10 meter resolution to 2.5 meter resolution. We use the UNet architecture to estimate the cassava or taro, the crop, the weight. By taking photo using the tablet PC, then the weight estimation will pop up. But you may be wondering why just to use a scale rather than these high techniques. But interestingly, there is no scale in Vanuatu. It's not technically no, but they have a really traditional bigger one. So also some scale using the battery kind of system. So battery is really expensive there. That's why we developed these applications. And using this application, we can collect any agro-meteorological data set. If we go to some very strong internet connectivity environment, then the monitored data will be updated to basically synchronized to the OSCA server to update the agro-meteorological database in the server. And we officially handed over and officially launched the OSCA system to Vanuatu government, and in front of the delegates from the Pacific Island countries, we officially launched the OSCA system in August last year. This is the kind of information delivery chain we designed. So we think about this kind of the supporting project for developing countries. Some fancy system, just they developed the system and handed over, that's all. So nobody used the system anymore after the project was over. So we really focus on how we make this system sustainable. That's why we designed this information delivery chain. Basically, we developed the trainers who will be able to train the neighboring farmers. This is the pictures. We train these guys. This is mainly government employees or some experts. And these guys train some key farmers. They selected the key farmers. Then these trained key farmers, finally they will be able to train their neighboring farmers. Okay. This picture shows I was really happy to find out this Facebook post. It's in November 2023. They are a government employee of Vanuatu. So without APC's help, they designed their own workshop to train these neighboring farmers. This is one picture of our system. So this post clearly show they are able to train the neighboring farmers by themselves, and also they are able to use the system by themselves. So we believe these pictures will give us some idea of sustainability. Okay, if you are interested, you can take using your smartphone, use this QR code, then it will give you some address to visit the OSCA system. Yeah. Oh, now we are in the middle of develop the second phase of Vancouver project. Okay. Actually, we started this developing back in 2022 December. The executive director of APCC visited Vanuatu and discussed with the Minister of Agriculture, also the Minister of Climate Change. And this NDA is really important to get the GCF project. Without NDA's endorsement letter, we cannot submit the proposal, any proposal. And also the AE accredited entity, the only accredited entity, AE, submit the proposal to GCF. This is how works GCF proposal funding system. So this is really important to get some support and endorsement from these governmental stakeholders. Last year, the COP28, anyway, they agreed to keep supporting to develop this second phase of Vancouver project. Last month, we visit again the Vanuatu. We meet the Minister of Climate Change and also the Minister of Agriculture. Oops. And this NDA. So remember, the NDA's endorsement letter will be crucial to getting the GCF project. Okay, and we visit some local office to conduct some crop experiment if we get the fund from the GCF again. Here's something different from Vancouver and second phase of Vancouver. And the first phase of Vancouver, the climate information is based on the province level. But we are designing to provide some community level climate information using the second phase of Vancouver project products. So for that, we developed these 6 components. The first 2 are getting some information. First one is some meteorological information from the ground-based observational network. The second is the OSCA system, second phase of OSCA system. We will, some more crops will be incorporated in this system. And third one is some interesting things. The first phase of Vancouver project, they are installing a single radar system. You may already know, using the radar system, we can get very accurate precipitation, the forecast sort of things. But the radar system cannot cover whole country. This SIA, this company has some techniques using some deep learning techniques and cellular imagery and the single radar system imagery. We can provide some whole radar-like images to covering the whole Vanuatu countries. So probably we can get more accurate precipitation forecast using this second phase of the Vancouver project product. Yeah, this is, and I'm sorry, this time a little bigger than the bigger front than the first phase. So we are asking we will ask maybe 20 million US dollar from the GCF. Okay, thank you, Thomas. This means thank you very much. Yeah, in Bislama. Thank you very much. Thank you, Thomas. Yeah.
예, 감사합니다. 우리 저희 질문 혹시 있으신가요? 질문 없어요? 네?
안녕하십니까. 저는 환경공학 전공하고 있고 뭐 약간 수처리 전공하고 있는 박사과정 학생인데요. 뭐 전공 관련 질문을 드리는 거 아니고 오늘 뭐 다 많은 오늘 발표해 주신 분들이 다 그 어떤 책상에서의 연구보다는 실질적으로 그런 연구들이 좀 뭐 전 지구적으로 아니면 우주적으로든 실제 적용될 수 있는 그런 연구에 대한 뭐 그런 결과들을 많이 보여주셨고 방금 말씀해 주신 내용들도 되게 그런 쪽으로 보여서 굉장히 의미 있고 좋은 일을 해주시고 계시다라고 일단 생각이 들었고요. 그런데 이제 저도 약간 그런 실질적으로 뭔가 연구를 적용하는 거 되게 관심이 많은데 이제 보니까 이제 연구들이 다 뭐 몇천만 달러 규모의 뭐 그런 일들을 하셨었고, 근데 그러려면 되게 많은 인력이 필요할 거고 또 시간도 많이 걸릴 텐데 그런 오스카 시스템 이런 거를 구축을 하고 또 실제 적용을 하기까지 어느 정도의 인력이 좀 필요했고 또 이제 어떤 어려움들이 또 있었을 텐데 어떤 부분들이 좀 어려운 점이 있으셨는지 그런 것도 좀 궁금합니다.
예, 정말 좋은 질문이시네요. 예. 사실 이 시스템 개발에 중점을 둬야 될 게 보통은 우리가 시스템을 디자인할 때 보면 기능 위주로 디자인을 합니다. 예. 그러니까 좀 더 많은 기능 그리고 뭐 유저 프렌들리라고 표현하는 그러니까 유저들이 사용하기 쉬운 그런 시스템을만을 생각하는데 근데 유저가 사용하기 쉽다라는 표현은 사실 두 가지 측면이 있거든요. 매우 간단하게 시스템을 구성을 하면 유저가 사용하기 쉬워지기도 합니다. 근데 요즘은 아주 복잡하게 시스템을 구성해 놓고 모두 다 이렇게 인캡슐레이트 한다고 그러죠. 예. 쌓아놓고 안 보이죠. 유저는 그냥 바깥에서 클릭만 하게 하는 그런 시스템을 만들기도 하는데 그러니까 우리가 주로 타겟하는 곳이 어디냐에 따라서 첫 번째로 가야 되냐 두 번째로 가야 되냐를 나눠야 되는데 이렇게 국제 협력을 통해서 개도국을 지원하는 사업에서는 저는 이제 첫 번째로 가야 된다고 봅니다. 시스템이 복잡해지면 복잡할수록 시스템도 무거워지고 또 거기에 그 인터넷 인프라를 생각해 보면 그런 그 무거운 시스템을 감당하기도 어렵습니다. 데이터가 아까 뭐 데이터 트랜스퍼링 하는 속도를 줄이기 위해서 그 텍스트만 트랜스퍼링 하는 시스템도 따로 만들었다고 했잖아요. 그런 것까지 고려해야 되고요. 사실 그리고 어려웠던 점이나 이런 거는 사실은 그렇습니다. 그러니까 이게 총 개발 기간이 한 5년 정도 걸렸던 것 같은데 처음에 프로토타입을 빨리 만들어내고 그걸로 계속 교육을 했습니다. 저희들이 바누아투에 여러 번 갔었거든요. 그러면서 이게 정말 그 아까 김문수 박사님이 말씀하신 버전으로 하면 아마 버전 10, 11 될 겁니다. 물론 이건 버전 1이니까 0.1, 0.2 이렇게 카운트해야겠지만 그런 식으로 했고요. 그다음에 실제 디벨로퍼들이 상당히 고통을 많이 받았죠. 여러 번 고치기도 했고 그래서 이제 저런 시스템이 겨우 나오게 됐습니다. 그래서 빠지기 쉬운 함정이 뭐냐면 내가 지금 개발하고 있는 시스템이 어디에 쓰일 것인가를 생각을 해야 됩니다. 그래서 그러니까 개도국에 간다고 하면 간단한 시스템으로 더 많은 사람이 사용할 수 있는 시스템을 구성해 주는 게 훨씬 효과적이고 그게 지속 가능한 시스템을 그나마 좀 보장해 줄 수 있습니다. 그리고 그 시스템이 이제 그 다음에 이제 버전 2, 버전 3로 가면서 점점 복잡한 기능들을 넣어주면 유저들도 점점 거기에 익숙해지니까 그 사용하는 사람도 더 늘어나고 좀 더 복잡한 시스템도 이제 잘 이용하게 되고 이제 그렇게 될 것 같거든요. 사실은 여기에 들어있는 기능들이 제가 뭐 자세히 설명을 안 했지만 지금 말씀드리면 되게 간단한 것들입니다. 근데 그런 기능 자체가 반도체에서는 처음이었어요. 예. 그래서 너무 고맙다는 이런 얘기도 많이 들었고요. 예. 그리고 지금 이 벤키라 프로젝트 전체 프로젝트에 아까 말씀드린 그 5개 키 섹터 중에 이 오스카 시스템이 그 성공 사례로 사실 뽑히고 있거든요. 예. 뭐 이렇게 이것저것 말씀을 드렸는데 예. 제대로 답변이 안 된 부분 있으면 다시 질문해 주시면. 예. 갑자기 눈물이 나려고 하네요. 옛날 생각이 나서.
아닌데 굉장히
네.
예, 답변 잘 주셔서 감사합니다.
예, 혹시 또 다른 눈물을 만드실 분 혹시 질문 혹시 뭐 간단하게 없으세요? 퀘스천? 제가 하나만 할게요. 에이펙 센터가 아는 분들은 아시겠지만 그 이렇게 학생들도 많고 그다음에 간단히 소개시켜주고 혹시 거기에 취직하려면 어떻게
예.
할 수 있는지 이런 부분들도 아마 관심 있으실 것 같은데 그거 간단하게
그전에 혹시 연봉이 얼마인지는 안 궁금한가요?
뭐 그거를 공개적으로 하기 좀 애매한 부분이 있어서 어쨌든 그 발표한 내용은 상관없는데 혹시 여기 관련 뭐 건사환하고 환생공 학생들 많은데 혹시 관심 있는 학생들의 정보가 될 수 있을까 싶어서 여쭤봐요.
예, 에이펙 클라이밋 센터 APCC 지금 뭐 부산에 있습니다. 지금 여기 그림에 나와 있는 거의 이 전경 그대로거든요. 예, 이게 국제 회의장이고요. 이 안에 들어가면 회의 시설이 갖춰져 있습니다. 그리고 아마 여러분 잘 아시겠지만 뭐죠? 영화의 전당, 그 부산 국제 영화제가 열리는 영화의 전당이 여기서 100m도 아니죠? 한 뭐 20m 이내에 있습니다. 그래서 매 10월에 되면 제가 처음에 그 APCC에 조인할 때는 그러니까 2012년인가요? 2012년 그때는 여기서 그 게스트들 있잖아요. 연예인들이 여기서 그 차를 타고 가서 영화의 전당에서 내리고 했었거든요. 그래서 우리는 일을 하면서 그런 걸 다 볼 수 있었는데 지금 위치를 좀 바꿨더라고요. 그래서 이제 그런 일은 안 일어나지만 아무튼 그렇습니다. 그리고 아마 연봉 같은 거 궁금해하실 텐데 입에 풀칠은 하고 살 수 있습니다. 그렇게 아시면 될 것 같고요. 그리고 여기 취직하면 하려면 어떻게 하냐 뭐 토익 보시고요. 주로 이제 아무래도 저희가 이제 클라이밋이라는 그 이름의 센터 이름에 클라이밋이 들어가서 기후 전공자들을 많이 주로 뽑긴 합니다. 저도 지금 하는 일이 계속 스포컴퓨터에서 기후 모델을 시뮬레이션해서 거기에서 나오는 뭐 수문학적인 성분하고 뭐 에너지 성분 같은 거를 비교하고 어떻게 하면 이런 성분들이 우리가 잘 시뮬레이션 해줄 수 있을 것인가. 근데 지금 우리에서는 이 수문 성분만 잘 맞추면 되는데 여기에 오면은 이 잘 맞춘 수문 성분이 어떻게 기후 시스템에 기여하게 되는지까지 이제 연구를 해야 됩니다. 그래서 좀 더 훨씬 더 글로벌한 그런 마인드를 가지고 계시고 기후에 연관된 그런 연구들을 꾸준히 하고 계시면 여기에 취직의 문이 열리지 않겠나 싶습니다. 근데 와서 그렇게 썩 좋은 직장은 아닙니다. 그러니까 꿈은 안 꾸셔도 될 것 같습니다.
예, 그럼 질문이 더 없으신? 저 지금 한번 뒤에.
네, 너무 잘 들었고 개인적으로 아까 질문하신 것처럼 뭔가 다른 대륙의 다른 그
네.
환경의 나라이기 때문에 우리나라랑 좀 다르게 고려해야 될 부분이 되게 궁금했었는데 그 부분에 대해서 되게 와닿는 답변을 들을 수 있었던 것 같습니다. 제가 좀 궁금했던 거는 아까 그 모델이 숏텀이랑 롱텀으로 그 클라이밋 체인지를 구분을 해가지고 어드레스
예.
할 수 있게 할 수 있다고 그 에임을 하셨다고 하셨는데 그 구체적으로 어떤 챌린지들을 에임하셨는지 궁금합니다.
예. 좋은 질문입니다. 그러니까 지금 우리가 이제 기후 변화를 얘기를 하지만 사실은 여러분도 아시겠지만 지금 우리는 기후 변화를 이미 겪고 있잖아요. 그래서 우리가 기후 변화라고만 생각하면 항상 기후 변화 시나리오만을 떠오릅니다. 그거는 이제 한 100년 미래를 얘기하는 거잖아요. 근데 이제 그게 이제 롱텀이 될 거고, 숏텀은 뭐냐면 지금 현재 우리가 겪고 있는 이상기후라고 불리우는 여러 뭐 홍수, 가뭄, 이게 예전과 다른 패런이나 뭐 그 프리퀀시로 발생을 하고 있잖아요. 그 현재 겪고 있는 그런 이상 기후들, 여기 이제 아무래도 숏텀이 되겠죠. 그래서 여러분 아마 그 날씨, 그러니까 기상과 기후를 구분을 하실 수 있죠. 예. 그렇죠? 예. 예. 그러니까 이제 기후는 아주 오랜 세월 동안에 있는 거기에 그 평균적인 그런 기후라고 표현하죠. 평균적인 날씨라고 해야 되나? 그래서 보통 우리가 비유를 통해서 하는 게 어떤 사람의 성격은 기후에 해당되고 아무리 사람이 온화한 성격을 가져도 어떤 날은 화를 낼 수가 있잖아요. 어떤 날 화를 내는 그런 느낌, 그게 이제 아마 기상과 날씨 이렇게 표현하는 건데, 그러니까 이제 그런 개념으로 생각하시면 되겠습니다. 롱텀은 기후에 가깝고 숏텀은 이제 기상, 날씨에 가깝다라고 생각해 주시면 될 것 같아요.
예, 혹시 또 질문 없으시면 예. 오늘 마지막으로 박수 한번 예.
감사합니다.
제 오늘 세미나 다 마쳤고요. 오늘 너무 끝까지 함께해 주신 참석자 여러분 너무 감사드리고요. 오늘 뭐 되게 다양하게 우주와 또 예, 센싱과 그다음에 또 다른 나라 굉장히 잘 못 들어본 나라에 대해서 굉장히 다양한 주제를 가지고 다뤘던 것 같습니다. 오늘 마지막으로 기념 촬영이 기념 촬영 제일 사실 중요합니다. 지금 이 부분이. 그래서 여러분들 가지 마시고 여기 단상으로 올라와 주셔서 마지막 기념 촬영 해 주시고 오늘 마무리하도록 하겠습니다. 예, 감사합니다. 예.